Hey, hello. This is a video about biochemistry, which is chapter 3 in our textbook. I just wanted to go over some of the major points in the chapter, just because we will go through it pretty quickly, and this is a way for people to come back and review the ideas. I'm going to go in this order. I'm going to follow this outline. I'll start with the first section with carbon compounds, and then we'll go into the second section, molecules of life. So the first bullet point is to distinguish between organic and inorganic compounds. Well, organic compounds contain carbon and other elements. If there's no carbon, it's not an organic compound. That's the easiest way to look at organic versus inorganic. Most of the chemistry we look at in biology will be organic chemistry. Carbon atoms, therefore, take a special role. Something to know about them is they like to have 4 covalent bonds, 4 shared pair of electrons. The bonds are shown as solid lines. We've done some drawings in class. So we have carbon-carbon bonds inside of here. Carbon bonds, some carbon bonds forming a ring. We can see a double bond here, or maybe even a triple bond. So we draw carbon bonds, or covalent bonds as lines inside in organic compounds. And they have all kinds of different shapes. And the shape of an organic compound is closely related to its function. In addition to the carbon in organic molecules, there are other atoms on the molecule. Sometimes these atoms are in groups that react together. There are 4 major groups we should be aware of. The hydroxyl group, which is represented as OH, it's an oxygen and hydrogen together, and for example, this is what it would look like on the edge of a molecule. We saw lots of hydroxyl groups when we modeled our sugars. Hydroxyl groups are going to make a molecule soluble in water. So that's an important thing to know. Anything with hydroxyl groups will be soluble in water. The carboxyl group, it's represented as COOH. Note the double bond in the oxygen. Sticking off the side here, we have a carboxyl group. It's also called a carboxylic acid, as they tend to make the molecules more acidic in nature. Amino group is NH2, a nitrogen and 2 hydrogens. Here we can see it, NH2. So we have an amino group. You may note that this molecule also has the carboxyl. These are the same molecule. We've just highlighted the amino versus the carboxyl group. It is okay for a molecule to have more than one functional group. Also note here that since this OH is part of a larger carboxyl group, it's not going to act as a hydroxyl. So there's a difference between the OH being isolated here and it being part of a larger functional group. It all depends on if it's connected to the same carbon as some other stuff. And then the final functional group for us is the phosphate functional group, which is this big thing with a phosphorus in the middle of it. It sticks out very obviously on the end of a molecule, and we'll see that in action with ATP. So these 4 functional groups, you should be able to recognize them on a molecule. You would not be expected to draw an organic molecule, but to recognize these 4 parts would be expected. Okay, so organic chemistry, we talk about small pieces called monomers and larger pieces called polymers. If you take 2 or 3 smaller pieces, small monomers, and connect them together, you get one polymer, or many mers. The process of doing that, the chemical reaction that does that, is called a condensation reaction. It's called condensation because you have water molecules are produced in the process as well. The process of splitting apart a polymer into its smaller monomer pieces is called hydrolysis because it requires water to react. To look at that as we did with modeling sugar, if you have glucose and fructose, these hydroxyl groups here, we can have an OH from one, the entire hydroxyl group from one, and just an H from another hydroxyl group. They can react with each other. With that reaction, when that reaction takes place, an oxygen stays behind. It becomes the link holding these 2 monomers together, and we get a water molecule out. That would be condensation, creating a larger sucrose polymer. It could be called a disaccharide. Saccharide means sugar, monosaccharide, disaccharide, polysaccharide. We could also take this water and run this reaction in reverse. If this water molecule was to break that bond in the sucrose, we could split the sucrose molecule into fructose and glucose. That would be a hydrolysis reaction, breaking molecules apart. This example here is given for carbohydrates, but the same idea of condensation hydrolysis would apply for any of the macromolecules of life, the large molecules of life, being carbohydrates, proteins, lipids, and nucleic acids. They're all built up by condensation reactions and broken down by hydrolysis reactions. So in addition to the four primary macromolecules, the carbohydrates, lipids, proteins, and nucleic acids, there's this one special molecule called ATP. That stands for adenosine triphosphate. It's abbreviated right here. So it's got a part, this big adenine part hanging out here on the side, and then three phosphate functional groups linked together in a chain. This makes it triphosphate. Closely similar or closely related to it is adenosine diphosphate, ADP. Here's the large adenosine and two phosphates next to each other. As you might imagine, we could take ADP, adenosine diphosphate, attach a phosphate onto it, and turn it into ATP, triphosphate. So diphosphate to triphosphate. In order to do that, we would have a hydrolysis reaction, the two hydroxyl groups reacting, making another bond inside, water being produced. This would be a condensation reaction making ATP. That chemical reaction, making an ATP, we're creating a chemical bond that wasn't there before. Note the bonds here, and note lack of bond there. Creating the chemical bond means we're storing energy in the reactants, in the product. So ATP has more energy in it than ADP. So this reaction, ADP plus a phosphate to make ATP, this is how your body will store chemical energy for later use. So ATP is an energy storage molecule. It's the primary energy storage molecule in your cells. If there's going to be energy provided to get some work done, chemically or physically, ATP is going to provide it. There's a larger view of that ATP molecule just to freak you out. You can see the three phosphate molecules inside of here bonded in. We have a sugar molecule in here and this adenine group with all these nitrogens inside of it. Again, what we need to know is ADP, adenosine diphosphate, plus a phosphate, condensation reaction occurs. We get ATP, and energy stored. Okay, so now we'll just take a look at each of the four macromolecules of life one at a time. The first one is carbohydrates. These are molecules that can contain carbon, hydrogen, and oxygen with a ratio of 2 to 1 with the hydrogen and oxygens. And we built this exact model in our lab with modeling sugars, glucose and fructose making sucrose. Glucose and fructose are monosaccharides, single monomer rings, so just a single piece. Since sucrose is two pieces put together, it would be a disaccharide. If we put more than two together, we get a polysaccharide. So monosaccharides making disaccharides, making polysaccharides, are making polymers of the carbohydrates. The major polymers of carbohydrates in plants, they would be cellulose, which their cell walls and wood are made of. And in animals, it would be glycogen, the jiggly stuff that you get around your belly if you don't have a six-pack. It would be mostly glycogen, stored also in your liver. So glycogen in animals, cellulose in plants, and the only difference is the way those molecules are put together. The shape of the polymer changes as you put them together. We could also note here that the primary function of carbohydrates in animal cells is energy storage. You store energy in the bond here between these, or within the actual ring of the monosaccharide itself. You store energy there, and then you break them down for energy later. In plants, that's also used for energy storage. Additionally, plants will use them for structure. The cell wall actually holds the plant up. So the function of carbohydrates and what they're used for. The next macromolecule of life is proteins. Proteins are polymers made up of amino acids. Amino acids, there are 20 different types of amino acid in the human body. The sequence of amino acids determines what the protein shape will be. We did the protein folding lab on this, where you saw the chemical properties of the different amino acids as they fold and twist around each other to make a more stable chemical situation for themselves. For example, some amino acids are hydrophobic, fearing water. Some are hydrophilic, loving water, wanting to be dissolved in water. So the hydrophobic ones will stay away from the hydrophilic ones. Twisting and moving of the amino acids from this primary sequence into this tertiary or third degree structure, which is the final shape of the protein. That final shape of the protein will determine what it does. Proteins are involved in all kinds of work and action inside of the cell, one of which is being enzymes. So enzymes are biological catalysts. They reduce the amount of energy needed for a reaction to occur, Enzymes are unique to a particular reaction. So there'd be some substrate, a chemical you want to break apart. The enzyme attaches to it and helps it break apart. It facilitates that, reduces the activation energy needed. Another way that enzymes can be involved is the enzyme might grab some reactant or two reactants and then also hold on to ATP in its other hand and take the energy from the ATP and transfer it to the reactants to kind of enable that reaction to occur by overcoming that activation energy. Almost any chemical reaction inside of the body has an enzyme that helps facilitate it or control it or regulate it in some way to make it easier. It's important to note that each enzyme is very specific. It will only work on a certain pair of reactants or on one molecule substrate. So this is where shape becomes very important. If, for example, you had two different sugars, so glucose and fructose, very similar, but their shapes are slightly different. An enzyme might attach to glucose and break it down, but not fructose. And that would affect the ability of you to digest one or the other of those sugars. The most common example is actually people who cannot digest lactose because the enzyme in their body, you need lactase enzyme to attach to the lactose sugar. If you don't have that enzyme, you can't break down that sugar. So the role of enzymes in chemical reactions to reduce activation energy, it's very specific. They call it the induced fit model. Okay, the third or next group of macromolecules in life are the lipids. Lipids are the fats and oils. They are all insoluble in water. Say they're hydrophobic. What defines a lipid? It's got a large number of carbon-hydrogen bonds. If you see, this is an example. Look at all the carbons and look at all the hydrogens bonded to them. There's a lot of carbons and hydrogens. There's another example. Lots of carbons and hydrogens inside of there. So they're nonpolar, they're hydrophobic, they do not dissolve in water. This is called a fatty acid. We have the fat part, which is just a long chain of carbon and hydrogen. That's what fat is chemically, large chain of hydrogen and carbon. And then there's an acid here. Note that functional group is the carboxylic acid. So we have a carboxylic acid with a fat. It's a fatty acid. This is also a fatty acid down here, but this one, this fatty acid, the fat part, has double bonds in it. So it's not all single bonds. Here, every carbon has all single bonds. It's completely covered with single bonds and surrounded by hydrogens. It's called saturated. If we have a double bond, meaning I don't have hydrogens in every single spot around the carbons, this is unsaturated, right? Saturated fats are going to be solid at room temperature because they stack up nicely and form a rigid structure. Unsaturated fats are going to be liquids or oils at room temperature because they don't stack nicely. They get these gaps in between because of the bends and kinks inside. These are fatty acids, one of the basic building blocks of other fats. If you take three fatty acids, and in this molecule here, I have three fatty acids that are all three of them saturated, and you connect them to each other using a glycerol, which is another lipid monomer. We have a glycerol molecule holding three fatty acids. That's called a triglyceride. Tri, because there's three fatty acids, and the glyceride, because there's glycerol there. Three fatty acids and a glycerol is a triglyceride. These are notorious for being in our food and making things taste really good and sometimes not be so good for us. Another type of lipid is a phospholipid. It's two fatty acids, long chain of carbon and hydrogens, connected to a phosphate group, this polar phosphate. Phosphate groups have all these hydroxyl groups on the outside of them. So the phosphate wants to dissolve in water. The fat does not want to dissolve in water, so we're half soluble, half insoluble. They form these double layers where the fat sides, the nonpolar parts, line up next to each other, and then the polar sides go on the outside, and you get this lipid bilayer, this double layer of phospholipids, which is the cell membrane, or the primary part of the cell membrane in cells. So phospholipids, super important. Two fatty acids, a glycerol, and a phosphate. The glycerol is the glue. So the difference between a phospholipid and a triglyceride is triglyceride has three fatty acids, and the phospholipid has two fatty acids, and then a phosphate group stuck onto the third link, which makes it soluble. Another type of fat is steroids. So steroids are fats. They're insoluble in water, but they are soluble in other fats, and that's what makes steroids good for sending signals across the body. They can just diffuse right through the cell membrane. Testosterone being an example of a steroid. Last group of organic molecules is nucleic acids. There's DNA and RNA. DNA is made up of a double helix. RNA is a single helix. DNA has has deoxyribose sugar. RNA has ribose sugar. That's the main difference. We have deoxyribose sugar in DNA. We have ribose sugar in RNA. These are the genetic information for the cell. They code for all the instructions on how to make things, specifically proteins in parts of the cell. We'll go into lots of detail between DNA and RNA. DNA is a double helix, RNA is a single strand, but it's the type of sugar that holds them together. That is our summary of biochemistry. Whoa, so much information, but it's all there. Hopefully this video helped you.